Once again, welcome to Kako Education Services, and our tutorials are being given by various teachers, and today's tutor is Nampa Cosmas. We are continuing with our topic of transport in animals. Previously, we looked at the mammalian cardiovascular system, and we said that it is made up of the heart, which is the pump, and blood vessels. Our tutorials have been majoring onto the blood vessels. And the blood vessels we have are the arteries, which carry blood away from the heart and take it to the respiring cells. Then we have the veins, which carry blood away from the respiring cells back to the heart. Then we have the capillaries, which connect arteries and veins. So during the transport of blood within the arteries, we said it reaches the blood capillaries where it delivers its oxygen and other materials where they exchange between the tissues and then the blood. After that, it gets back to the veins where it is transported back to the heart. Blood plasma and tissue fluid. Blood is composed of cells, and these cells are floating in a pale yellow fluid which is known as plasma or blood plasma. Within this blood plasma, we have various substances like glucose, plasma proteins, waste products like urea. We also have sodium ions, hemoglobin. 90% of the blood plasma is majorly water. As blood flows through the capillaries within the tissues, some of this plasma leaks out and seeps into the space between the cells and the tissues, forming a fluid known as the tissue fluid. This tissue fluid is almost identical in composition to blood plasma, implying that they almost have the same composition, though some of the substances that are in blood plasma are not present in the tissue fluid. The few substances that are not present in the tissue fluid are the protein molecules, and this is because they are large to pass through the tiny holes in the capillary endothelium. The tissue fluid also does not contain red blood cells, and the reason is that these red blood cells are too large as well to pass through the tiny pores within the endothelium of the capillaries. Some white blood cells are capable of squeezing through these tiny pores and move freely within the tissue fluid. In our subsequent tutorials, we are also going to talk about the white blood cells, and we are going to look at their composition and what the different types that we have for the white blood cells. So, the composition of the tissue fluid and blood plasma is almost the same, like we have said. The differences are in the substances, but also in the concentrations. For example, when you look at the concentration of water in blood plasma, it is higher than that in the tissue fluid. The concentration also of sodium ions varies. The concentration of urea also varies greatly. You may be wondering what exactly brings about formation of tissue fluid. We have to know that as blood leaves the aorta, it is pumped with high blood pressure. And it is this blood pressure that will get into the capillary bed that will be responsible for pushing out the fluid into the capillary endothelium, then to the tissue space. Thus, we can say that the pressure inside the capillary bed is enough to push the fluid out of the capillary into the tissue space. However, we have to know that a concentration difference in terms of ions between the tissue fluid and blood plasma. The blood plasma contains higher concentrations of ions, and then the tissue fluid contains low concentrations, which means that water drains from a region of higher concentration to the region of low concentration. That is in terms of water molecules. It is going to move from the tissue fluid back to the blood plasma. With these two opposing movements, whereby the tissue fluid is moving from blood plasma into tissue space, and then water is moving from the tissue fluid into blood plasma, it brings about a certain gradient, whereby at the beginning of the arterioles, the tissue fluid is going to be pushed out of the blood plasma. And then at the beginning of the venous, we shall have water joining the blood plasma so that it continues its way within the cycle of the blood saturation. If the blood pressure in the arterioles is high, it implies that much of the blood fluid is going to be pushed out of the capillaries into the tissue space, which will cause an accumulation of the tissue fluid, resulting into a condition known as oedema. This oedema condition can be controlled by the reduction of pressure in the arterioles. When the pressure in the arterioles reduces, little fluid is pushed out of the capillary bed, and therefore we shall have no accumulation of the tissue fluid within the tissue space. The tissue fluid forms an immediate environment of each of the individual body cells. And it is through this tissue fluid that the exchange of materials between cells and blood occurs. Within our bodies, so many processes take place to maintain the composition of tissue fluid at a constant level to provide an optimum environment in which the cells can work. You may ask yourself what happens to the fluid when it gets into the tissue space. About 90% of this tissue fluid leaks back into the capillaries and joins the blood plasma. The remaining 10% is collected and returns to the blood system by the means of a series of tubes that are known as the lymph vessels or the lymphatics. That is how the 10% is going to join the blood system, using the lymph vessels or the lymphatics. These lymphatics or lymph vessels are tiny blind ending vessels which are found in almost all tissues around the body, so that they can move the remaining 10% of the tissue fluid. The lymphatics or the lymph vessels contain tiny valves within them to allow the tissue fluid flow in them, but stop the fluid from flowing out of the lymph vessels. Or we can say that these valves allow the fluid to flow into the lymph vessels, but do not allow it to leak out back to the tissue space. These valves are similar to those that we have talked about within the veins, maintaining one direction flow for the fluid. The valves in the lymph vessels are wide enough to allow large protein molecules that could have passed through the tiny pores in the capillary endothelium to be carried within the lymph vessels. If these large protein molecules are allowed to accumulate around the tissues and then the capillary bed, it would bring about a condition that we had already discussed, which is oedema. When you have a large concentration of these protein molecules, much of the fluid is going to move from the blood plasma into the tissue fluid due to the high concentration of ions which is present within the tissue fluid. And this in turn is going to bring about an accumulation of the tissue fluid that is going to cause oedema. That means the valves as well stop the linking of the blood proteins back to the tissue space. The fluid which is already inside the lymphatics or the lymph vessels is no longer referred to as tissue fluid, but instead it is called lymph. We call this fluid lymph, and it is identical in terms of composition to the tissue fluid. That is, they have the same composition. The materials which are in the tissue fluid are the same as the materials that are going to be within the lymph fluid. In some tissues, you will find that the tissue fluid has a different composition to the lymph fluid. For example, within the small intestines. After a meal, you'll find that the lymph vessels have a fluid that has a large composition of lipids that are going to be from the digested food. And these are going to be different from the tissue fluid that is around the blood capillaries that are present within the small intestines. Also, when you look at the lymph that is within the liver, you'll find that there is a high concentration of proteins that is going to be present in the lymph. To sum it up all, you'll find that the compositions of the tissue fluid and the lymph, in some tissues it is the same, and in some tissues the composition is different, slightly different, just because of the materials that are present in them at that time. The lymphatics join up to form a large lymph vessel, and these gradually form a transport system. They transport the lymph back to the large veins, that is at the subclavian veins, where they will join the lymph to the blood system. The movement of the fluid along the lymphatics is largely caused by the contraction of muscles around the vessels and keeping going in the right direction of the valves. So you realize that here it is more similar to the movement within the veins. Just the contraction of muscles creates the pressure that is going to push the lymph up to the subclavian veins, where the lymph now joins the blood system. Along the lymph vessels, there are lymph nodes, and these are involved in protection against diseases. Within the lymph nodes, there are some of the white blood cells which produce antibodies that are capable of killing bacteria and removing pathogens. Therefore, some unwanted particles and bacteria are removed from lymph as it passes via the lymph nodes that contain these white blood cells. To conclude this tutorial, we have been able to talk about tissue formation, which is as a result of blood pressure that is within the capillaries and having these pores which allow the fluid to move from blood plasma into the tissue space to form the tissue fluid. We have also discussed that the tissue fluid gets back into the blood system, either at the venous end or by use of the lymphatic system that is going to join the subclavian vein, and then the lymph or the tissue fluid joins the blood system. We have also been able to note that along the lymphatic system, we have the lymph nodes that contain white blood cells which remove unwanted material and bacteria from the lymph fluid. And we have said that this lymph fluid is from tissue fluid, whereby the tissue fluid which remains in the tissue space, and it makes about 10% of this, joins the lymph vessels, and once it is already in the lymph vessels, it is no longer called tissue fluid but rather lymph fluid. Thank you very much for listening. Subscribe.